Welcome to this module on image analytics and biophotonics. In this session, we'll examine the significant role image analytics plays in advancing modern healthcare. Our focus will be on the integration of biophotonics and artificial intelligence, highlighting essential imaging techniques, cutting-edge processing methods, and practical real-world applications. Together, we'll uncover how these advancements are revolutionizing diagnostics, therapies, and the development of personalized healthcare solutions for the future. In this presentation, we'll explore how image analytics is evolving, especially in the context of healthcare and biophotonics. We'll begin by grounding ourselves in the fundamental principles of image analytics, then move into practical workflows and how they're applied in biophotonics. From there, we'll take a closer look at how AI is being used in medical imaging and examine a variety of imaging techniques, highlighting success stories where AI has made a real impact. We'll also address some of the challenges faced when deploying AI in medical imaging and consider what lies ahead for data-driven technologies in healthcare and beyond. Finally, we'll summarize the key takeaways from the session. Here, we focus on understanding the fundamental principles of image analytics. At its core, an image is essentially a matrix of numbers, with each number representing a pixel or picture element. These pixels are the smallest units of an image, and their arrangement creates the visual representation we see. It's important to understand that the edges between these pixels are artifacts of imaging or digitization. They don't physically exist. One of the key tools in image analytics is the histogram, which shows the distribution of pixel intensities in an image. This distribution reveals critical information about the image's brightness, contrast, and dynamic range. For example, a narrow histogram indicates low contrast, while a broader histogram suggests a wider range of intensities. Moving into practical techniques, we begin with pre-processing, which involves steps like noise reduction and normalization. Noise reduction, often performed with Gaussian filters, eliminates unwanted artifacts that degrade image quality. Normalization, on the other hand, adjusts intensity values to create uniformity, making the image more suitable for analysis. After pre-processing, the next step is feature extraction, where meaningful information is derived from the image. This process includes identifying edges to define object boundaries, analyzing textures to recognize patterns, and quantifying intensities to assess brightness and contrast levels. Together, these concepts, pixel-based representation, histogram analysis, pre-processing, and feature extraction, serve as the building blocks of image analytics, paving the way for more advanced applications in healthcare and beyond. This slide outlines the essential components and techniques used in medical image processing, providing an overview of the stages from image acquisition to visualization. Image acquisition. The process starts with capturing medical images using techniques like MRI, CT, or fluorescence microscopy, which forms the basis for further analysis. Pre-processing. Removing noise and artifacts using filters like Gaussian blur, resampling and cropping data for efficiency and ease of processing. Segmentation. Identifying and isolating anatomical regions using methods such as Gaussian blur, Otsu's threshold, connected component labeling. AI-driven machine learning algorithms are frequently applied for advanced segmentation tasks in medical imaging. Registration. Aligning multiple images or datasets from different sources to ensure consistency and comparability. Feature extraction. Extracting critical details such as boundaries, edges, and textures, statistical measurements like center lines for specific regions. Classification. Using machine learning models to categorize features and diagnose conditions based on image data. Visualization, rendering processed images for interpretation and exporting models for simulations, CAD designs, or even 3D printing of anatomical structures for further study. This workflow demonstrates how modern image analytics integrates traditional processing methods with AI innovations to deliver accurate, actionable results in medical diagnostics and treatment planning. Now let's see how AI supports and enhances different stages of the image analytics workflow in medical imaging. In classification, AI models can be trained to identify and differentiate between multiple disease types. For example, in retinal imaging, deep learning architectures like VGG16 or Inception v3 can distinguish between conditions such as choroidal neovascularization, diabetic macular edema, drusen, and normal retinal tissue. Each classifier is trained for a specific condition, and their combined output enables multi-class classification with high accuracy. In segmentation, AI helps isolate and delineate specific anatomical layers or structures within an image. Here, we see two different segmentation approaches, patch-based and semantic segmentation. These methods are used, for instance, to segment retinal layers from OCT images, allowing for detailed layer-by-layer analysis. The use of filters and probability maps, followed by AI-based training, allows for precise tissue mapping and classification. Next is image recovery and reconstruction. AI models, especially convolutional neural networks and UNets, are used to refocus defocused images, remove blur, and even reconstruct data from limited inputs. These neural networks can be trained to optimize LED illumination patterns or perform cross-modality recovery, enabling accurate reconstruction even from noisy or incomplete images. Lastly, GAN-based computational H&E staining represents a powerful application of AI in pathology. Generative adversarial networks can simulate traditional hematoxylin and eosin staining digitally, generating virtual stained images directly from label-free input data. This not only speeds up the diagnostic process, but also reduces the need for chemical reagents. These examples illustrate how AI plays a critical role in making image analysis more accurate, faster, and scalable across different domains in medical imaging. Moving into some of the most commonly used imaging techniques in biophotonics as illustrated on this slide. These methods are essential tools in modern diagnostics, research, and therapeutic applications, each offering distinct capabilities for specific needs. On the left, we see techniques like optical coherence tomography, OCT, which generates high-resolution cross-sectional images of tissues, making it invaluable in fields like ophthalmology and dermatology. Raman spectroscopy imaging, another critical method, enables chemical mapping at the molecular level with applications in cancer detection and drug analysis. Confocal microscopy provides high-resolution imaging by focusing on a single plane within a sample, proving especially useful in cellular and molecular biology. Structured illumination microscopy, SIM, enhances spatial resolution beyond the diffraction limit, making it ideal for detailed cellular studies. On the right, fluorescence imaging stands out as a versatile technique for visualizing specific molecules within cells or tissues, while multiphoton microscopy facilitates deep tissue imaging with minimal photo damage. Thermal imaging, or infrared imaging, is widely applied in detecting temperature variations for medical diagnostics, such as inflammation identification. Lastly, holographic imaging captures 3D information about a sample, offering innovative approaches to both diagnostics and visualization. Together, these diverse techniques, complemented by advancements in AI and computational tools, are revolutionizing how we capture and interpret biological data, paving the way for precision medicine. This slide highlights an advanced application of imaging techniques enhanced by AI, optical coherence tomography, OCT, in ophthalmology. OCT is a foundational imaging modality, providing detailed cross-sectional views of the retina essential for diagnosing various eye diseases. AI-powered OCT systems have revolutionized this field by offering several key benefits. Early detection. AI identifies subtle retinal changes, enabling early diagnosis of conditions like diabetic retinopathy and glaucoma. Automated analysis. These systems deliver consistent and objective evaluations, reducing variability among clinicians. Enhanced accuracy. AI-driven analysis improves sensitivity and specificity, even for detecting rare pathological signs. Real-world applications. Platforms like Altris AI are pioneering innovations in ophthalmic diagnostics. Efficient workflow. By streamlining image interpretation, AI supports faster and more informed clinical decisions. As depicted in the GIF on the left, millions of OCT scans can now be systematically labeled and analyzed using AI. This capability enables differentiation of disease severities and identification of a wide range of pathologies. The integration of imaging and AI is transforming diagnostic precision in ophthalmology, setting new standards for patient care. Let's look at two leading examples where AI is making a transformative impact in medical imaging: Zebra Medical Vision and Path AI. Zebra Technologies has developed AI tools for radiology that can detect multiple diseases from a single scan, increasing the diagnostic value of routine imaging. Their system also enables retrospective screening, such as identifying osteoporosis from existing CT scans, revealing health risks that might otherwise go unnoticed. The platform integrates smoothly into hospital workflows through lightweight agents that connect to the cloud for model updates, ensuring minimal disruption. It delivers consistent performance across various imaging systems and patient populations, and focuses on conditions that are underdiagnosed yet have a major impact on public health, often referred to as silent epidemics. On the pathology side, Path AI is driving innovation by automating time-consuming diagnostic tasks. Their system analyzes tissue images and integrates genomic data to inform therapy decisions. Using machine learning, it identifies novel cancer signatures, classifies cell types, tumor grades, and genetic mutations. The goal is to discover new features with predictive value for treatment outcomes and survival. Path AI's software is designed to exceed current standards in diagnostic accuracy and consistency, supporting clinicians with deeper insights for more personalized care. These examples highlight how AI is reshaping medical imaging, not only improving accuracy and speed, but also enabling new diagnostic and therapeutic possibilities at scale. As we've seen, AI has strong potential to revolutionize medical imaging, but bringing these innovations to real-world clinical settings involves a number of critical challenges. These barriers must be addressed early on for companies to develop scalable, trusted, and commercially viable solutions. Technically, many AI models still struggle with inconsistent results. A single input can yield different outputs depending on when or how the model is used, making predictions unreliable. Without mechanisms for detecting unfamiliar or out-of-distribution data, models may give confident but incorrect outputs, often referred to as silent failures. Poor calibration and lack of explainability further limit clinical usability and regulatory approval, both of which are essential for commercial success. When it comes to real-world deployment, things get even more complex. AI systems must handle images from different populations, cameras, protocols, and varying biological conditions. Models trained in one setting may not work well in another without adaptation. What works in a controlled environment doesn't always translate smoothly into busy, diverse clinical workflows. These are not just technical issues, they are business-critical factors. From a commercialization standpoint, failure to generalize or adapt to local contexts can block adoption. Most diseases are not black and white. They exist in a diagnostic gray zone that requires models to make nuanced decisions. If a system overfits or carries hidden biases, its value in real-life settings drops significantly. Additionally, the lack of continuous monitoring can make it difficult to track performance over time, a key requirement for long-term trust and compliance. Recognizing and tackling these challenges head-on allows companies to design better, more reliable products from the beginning. For any health tech company aiming to bring AI imaging tools to market, these considerations are vital, not just for technical performance, but for building trust, meeting regulations, and scaling a sustainable business. This slide highlights the future directions for advancing data-driven technologies in healthcare and beyond. The integration of AI and data science presents enormous opportunities, but it requires a structured approach to ensure efficiency, reliability, and ethical considerations. Standardization across modalities is crucial. By developing unified protocols for data acquisition and analysis and adopting the FAIR principles, findability, accessibility, interoperability, and reusability, we can reduce variability and improve reproducibility across studies. Ethical data sharing and collaboration emphasizes the need for frameworks that foster multi-institutional cooperation while ensuring data privacy and compliance with regulations like GDPR. Federated learning is emerging as a key solution, allowing institutions to collaborate in training AI models without sharing raw data. Synthetic data for model training is transforming the landscape. Techniques such as generative adversarial networks, GANs, can create synthetic data sets, helping expand AI's applications to rare conditions and addressing concerns around data bias and quality. Next, open science and innovation models embrace transparency and cross-field knowledge sharing. By leveraging external expertise and open innovation frameworks, we can enhance reproducibility and protect intellectual property. Cross-disciplinary inspiration encourages us to draw from fields like genomics and proteomics to manage and integrate large data sets effectively. Techniques like site effect removal and multi-site data integration are paving the way for better analysis across imaging and sensing technologies. Real-time AI-driven diagnostics focus on providing actionable insights during medical procedures, improving decision-making, especially in personalized diagnostics and remote monitoring. Finally, robust validation frameworks stress the importance of rigorous pipelines to ensure reliability in clinical and safety-critical environments. Incorporating uncertainty quantification techniques can assess confidence in AI predictions, ensuring trustworthiness in its applications. Together, these priorities lay the foundation for fully leveraging AI and data-driven technologies, shaping the future of precision healthcare and beyond. This slide wraps up our session on image analytics and biophotonics with key takeaways. Biophotonics imaging is transforming diagnostics by enabling precise disease detection and advanced image reconstruction. These technologies provide non-invasive, high-resolution insights that are critical in modern healthcare. Artificial intelligence plays a vital role here, not only by enabling real-time analysis of complex medical images, but also by generating synthetic data for training models and fostering innovation through interdisciplinary collaboration. Success stories like PathAI, Zebra Medical Vision, and AI-powered optical coherence tomography show us how these technologies are already making a real-world impact, improving diagnostic accuracy and clinical decision-making. However, deploying AI in clinical environments brings challenges. We must address not just the technology, but also regulatory, clinical, and integration hurdles to ensure scalable and reliable adoption. Looking ahead, the future depends on standardization, strong validation processes, and ethical data sharing. These are essential to ensure reproducibility, trust, and collaboration across healthcare and research ecosystems.